Halo, pagi Pak. Pagi Kak.
Iya, ya. Halo, Vincent ya?
Mm-mm.
Oke. Iya, jadi
Enaknya panggil apa Kak? Kak Bima?
ya Kak Bima aja nggak apa-apa. Jadi aku ini aja apa namanya, ngikutin apa, maksudnya kayak gimana kalian pengennya gitu. Maksudnya
Nah,
aku bisa mulai kapan aja.
oke.
Ini presentasinya mau pakai bahasa Inggris apa Indonesia?
Up to you sih Kak.
Oke, oke. Ya, maybe it'll be easier to talk in English because
Hmm.
I don't know, like maybe it's a good practice as well
Hmm.
because yeah.
Oh ya, Verlin sudah direkam belum ya? Izin direkam ya Kak?
Boleh, boleh.
Yes.
Iya, aman-aman.
Usually Kak, we will present our material for like maybe 10 to 15 minutes about
Mhm,
basically overview of our project, and
mhm.
then what's been going on with our project, and also the challenge that we encountered in our project. But usually before that, um, the speakers present first
Ya.
about the material that we want to hear.
Mhm.
From you, Kak Bima, actually, we would like to hear about protein engineering, especially utilizing AI, which what you've been doing with in the Baker's lab, right?
Oke.
Using the
Ya.
de novo pipeline. So
Mhm.
I think that's what we want to hear first.
Okay. Yeah.
Yeah,
So
good.
I think that sounds good to me. Well, I prepare like some slides basically.
Oh, that will
But
be perfect.
yeah,
Yes.
yeah, but I mix between basically DNA nanotech and the novel protein design because I just don't want to. I mean, I did my PhD in DNA nanotech, right? So I also want to include that basically, and so that you guys can also like know about that field, and then protein design I also like present. Maybe not that into like very technical detail, but it will be just like introduction and a little bit of like survey. And for follow up, you can always like send me email if you have questions. We can schedule, let's say, another session because I saw like your proposal basically, your team proposal, and then I don't directly work with antibody or like nanobody, but I mean, I'll try my best to be a good resource, basically. So yeah, I mean, we can always discuss, like now and also like, like in the near future. Yeah. So yeah, I'll share my screen. So my voice is clear, right?
Yes.
Okay.
Yes, Kak.
Okay.
All right. We also can can see your screen right now.
Okay.
Yes.
Yeah, it's a bit weird in the for the Teams. This is Google Meet, right?
Yes. Is something new for you? Google Meet.
No, it's just like, how do I make it like in the presenters mode, like show the slide basically? Or maybe I need to do this. Um, is it good now? Like, do
Yes.
you guys see like the full screen?
Yes.
Okay.
All good.
Yeah. So as I said before, this more will be like only like like introduction and a bit of survey so that the team can be aware of these two fields. I don't want like to pick favorite over this because I do enjoy both of them because I did my PhD in DNA nanotech, and then now I've been working in the Baker lab for about 5.5 months. So pretty recent basically. So I'm also like still learning about the novel protein design. So yeah, I mean, it's not like, like I'm in a baker lab and then I will be like, like a master of protein design, but like we keep learning basically, right? So yeah, and then we will discuss. Um, a little bit about myself. So I did my undergrad in ITB in engineering physics. So nothing about biology, molecular biology, biochemistry, nothing like that. And during that time, I had a chance to intern at CERN, basically in Switzerland, Geneva, where there I did some project about particle detector for high energy physics. I also did in the internship towards my senior year during my undergrad uh, in Japan at Tokyo Tech, yeah, in Japan, where I did semiconductor, nanotransistor, nanoelectronics, and then that basically kind of like brought me into my master study in material science and engineering at UCSD in San Diego, California. And then I switched or transition into biophysics and physics with um, uh, Rizal, where I did basically mostly biophysics, single molecule experiments using DNA nanotechnology to study dynamics of single molecule under tension. And now, like since January this year, I joined Baker Lab at UW in Seattle, in the Institute for Protein Design, where I work on protein motors, trying to basically build the Novo protein motors, which is very different to what like existing protein motors that we have in nature. So to get started, I always wonder with living systems that what makes me basically like transition from physics, engineering physics, to a more a biology friendly field, which is now in protein design. So in living systems, for example, like our cell, we have like many uh, functional biomolecules where they interact with each other. They're like basically nucleic acids, proteins, lipids, sugars, and these molecules, they have their own functions, which basically like makes living system, like can function. And the question is, out of like, like we have like those interactions which are like great, but can we design the novel biomolecules which have new functionalities or like outperform the existing ones? So basically, that's kind of like one of the motivation that drive me towards like um, going into nanotechnology field and now like protein design, as we know that protein is the machinery of living systems. So everything that undergoing like in living systems, basically like like proteins, they proteins do the job, and that's basically motivates me like like can we design something so that we can outperform these existing ones. To start with, um, I think many of you know about the central dogma in biology, where the flow of information, genetic information, flow like from DNA, and then transcribe it into mRNA, and then mRNA will be like basically translated into amino acid sequence, and then this amino acid sequence, depending on the sequence, it will spontaneously fold into specific 3D protein shape, which basically will govern the function of the protein. And as I said before, the fields that I have been working with, basically like DNA nanotech and DNA and the novel protein design, these two fields basically sits right at the heart of this central dogma, where DNA nanotechnology tries to utilize this Watson-Crick base pairing to create new structures of DNA, functional structure of DNA that can perform several functions. While for the novel protein design, it tries to generate new protein backbones that have never existed before in nature for new functionalities. And I will start with DNA nanotechnology first, just a little bit of survey. So in DNA nanotechnology, I think the field started like around like in the 90s when Nat Simon, he basically tried to make this DNA crystal, 3D DNA crystal, and then basically his research group uh, try using these building blocks of four-way junctions to just to create like a simple 2D lattice. So here you can imagine like if you have like this DNA, different four DNA sequences, which can hybridize because of using the base pairing. A basically pairs with T and then G pairs with C. And then you design overhangs in each arm so that when you mix all of them together, it will just grow into a 2D lattice through self-assembly. And you can imagine you can design different shape of this tile, for example, like this DX tile or like Y-shaped tile to achieve a different shape of this 2D lattice. And then in 2006, basically, I think about 2 decades ago, Paul Rothman, he invented DNA origami, which is very clever because now you don't have to design these tiles um, 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 manually. So you can now just like fold a very long DNA strands. Let's say here you can see the gray strands are like the ones that are being folded by many short staple DNA. So this long DNA strand, the gray one, this is can be basically derived from bacteriophage DNA, M, basically M13, MP18. And then this basically long DNA strand, it has unique sequence. And then we can design short staple strands that can connect different basically points in this long DNA strand so that it can fold to a specific design structures. And with this basically technique of DNA origami, people have shown that they can basically make many shapes of nanostructure. As you can see here, like from the design on this row, and this is the experimental data from AFM, where you can see the agreement between design and the experimental data. And it's not only about doing 2D shapes. You can even like go crazier with 3D. And technically, the limit is just like your imagination and the length of this long DNA scaffold. So here, for example, you can make like sort of like, like a brick shape or, like a curved shape like this, um, which, like very amazing because, like previously it was like very hard to make this nanostructure with DNA. And some application about DNA origami, again, you can always like read more papers about it, but people in the DNA nanotech field, they use DNA origami for different purposes. One, you can imagine that this DNA origami as a platform where you can attach different molecules or like different therapeutics modalities. For example, you can attach, or like you can add doxorubicin in, which will intercalate basically in the groups of the DNA, which can be like a therapeutic agent here. Or like you can also like add therapeutic nucleic acid, which can, let's say, like silence any genes that you want to silence. Or you can basically, like dock different types of antibodies or like proteins that can interact with the target of interest and then activate or like affect the downstream signaling. And people have basically developed this for several applications such as vaccines or like for tumor applications. And one interesting case is like where they developed this DNA nanorobot, basically, where it will only open when it finds the target of the antigen target, and then it will basically open the cargo, and then the antibody fragment can be exposed to the target cell, basically, and then it will affect or like like help with the therapeutics. So that's some overview about DNA nanotech. So if you are interested more about applications or like more details about it, you can ask questions later and we can discuss. And then now, what I would like to uh, discuss is the second one about the novel protein design. So this field basically, I think, started with David Baker in the 90s or like late 90s, and then I think the first de novo protein was um, basically expressed or like successfully expressed in 2003, I think, when he published his basically like groundbreaking paper. The idea is like in the conventional protein engineering or like like science discovery. Usually, we know the sequence. We know like basically like the sequence of the gene, and then we know like the sequence of the amino acid, and we know the protein, and then the biological function. So basically, like it starts from like the existing protein in nature, and then we characterize, oh, this protein has this A function, or the other protein has B function. So it's more like trying to understand what nature has basically provided us with. But in the Novo protein design, the paradigm is shifted uh, backwards, basically, like where we start with a desired function, and then we try basically to find a protein backbone that can accommodate or like do that function. And then we try to design sequences or like a sequence that can fold into that design's protein backbone. And then we order the gene and try to express it in the wet lab. And about protein design, it was a very hard problem because protein is not as simple as DNA. So many of us, I think, already know like DNA, there are like 4 bases basically, right? And then the pairing is clear, AT-CG, and it's basically only involves a hydrogen bond and pi staking. But in protein, you have like 20 amino acids, and the interactions can involve basically hydrogen bond, ionic interaction, van der Waals, pi interactions. So those are like basically like making, like to predict protein from its sequence is very challenging. So I think this protein prediction problem or protein folding problem had been there for like quite several decades until I think in the 2020 or like 2019 when like AlphaFold is released basically based on like like a deep learning a model that can predict protein structure from sequence. So right now, like basically nowadays, there are like several protein prediction model that we have. One of them is like AlphaFold. Baker Lab also developed RoseTTAFold. There is also like chai, which is specific for antibody, and chai is basically like, I think I've never used chai, but I think it's based on like a company. So we can now basically like predict from sequence, amino acid sequence, into protein 3D structure with high accuracy. I mean, it's not like 100% accuracy because it's just prediction, but like compared to like several decades ago, like like it's nowhere basically. It was nowhere as compared to now. And the challenge also like in protein design, other than like predicting what we design, is to find the sequence that can accommodate the backbones that we design. So first we have to design the backbone basically, which is challenging because like it's not like DNA where you have like a double helix, right? Clear. But with protein, you have secondary structure, and then this secondary structure, let's say like alpha helices, beta strands, it can basically like interact in 3D and then form like a vast majority of a vast arrangement of this beta helix alpha strand, and also like you have loops. So that's the problem. So that's and to basically like find the sequence that can fold in this basically like vast majority of like um, landscape of folding. So we have like so many local minima. So we have to basically like sample many um, many possibilities in the past. But now like with this deep learning like basically model, we can get the sequence into structure with relatively high accuracy. And yeah, that's why like I think they got like Nobel Prize in 2024 because this is like a very long standing problem, and then they made it basically like possible to design protein de novo, which is like like new, like non-existing before in nature, and predict this with high accuracy. And why do we basically do like the novel protein design? So let's just like take an example of a, a protein with 100 residue. So because we have like 20 amino acids, so it means like we have this possibilities of like basic a number of like protein sequences. But in nature, we only observe like 10 to the power of 15 sequences basically, like, and 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 what we want basically like to explore more is like this space that nature hasn't provided us with what they basically like have in nature. And traditional protein engineering basically they start working with like whatever like existing backbones that we have in nature, and they start to do like basically modifying this natural backbones with small mutations with to improve the functions or like the performance of the protein. But in the Novo, we basically like make the backbones from scratch, and then we try to find sequence that can accommodate that backbones, and then characterize it in the wet lab basically. Yeah. And with AI, like basically like I think after 2020, after AlphaFold, Baker Lab also like shifted their like computational method from physics based that they used to use basically using Pi Rosetta into this deep learning basically like using all the protein structures in the PDB data bank, and then use that PDB data to train a model so that they can generate de novo backbones that can do some functions that we want. Yeah. So this is like some examples of what they have achieved. These are like not my work as a disclaimer. So if you're interested more, I can send you these papers. But yeah, they basically like showed uh, they can design binders to an insulin receptor with high affinity, as well as to this influenza target, and also to this immune receptor target. Yeah. And how they generate protein backbone with AI? Basically, they use a diffusion model. So this diffusion model is it's already like very successful in image generation, such as like DAL-E, where or like stable diffusion and DAL-E. So where let's say like they train the neural net model with a bunch of images. Let's say like like they train with many images of cats, and then those images they basically add noise to it. So they basically have, let's say, like a training set where they start with cat and then they give noise. You move like from right to left, and then you in the end like you get like a whole Gaussian noise here. So at each step they basically crop the image more and more until they get like just like purely white Gaussian noise. And then they train a neural net to denoise this basically like white Gaussian noise from different time point to get back into their training set. So basically like first they add noise, and then when they basically like like have this training set, they try to denoise this no basically corrupted images to get back to the training data. And then by doing this with the same principle with protein, where let's say like you started with any protein structure in the PDB data bank, and then you add Gaussian noise from right to left, and then after that you train the neural net to basically denoise this like Gaussian noise to get a completely a new protein backbone. So by doing this, basically at each step, because you are like trying to denoise it, so there will be like some kind of like new stuff emerge, and then you will get completely a new protein backbone as compared to what you use to train the model. So it's not like completely when you denoise, the model will completely denoise into the exact protein backbone that you used to train, but it will basically add some kind of like diversity in the backbone so that it will be different than what the training data set. So that's why it's a de novo backbone. And they published this in 2023, so like way before like I joined the lab basically. But this is basically like the bread and butter of the lab, and they basically develop a newer version of RFdiffusion, which is RFdiffusion 3 or RFD3, where now it's all atom. So previously this RFdiffusion 1 or like RFD or RFdiffusion only, it basically works without all atom formulation. So it only like basically works with backbone atoms, and then later like MPNN assigned the atom the residues, and then the atomic position will be inferred from that sequence basically. But then by simulating or like by working with all atom model, we can do much more for the Novo protein design. For example, many tasks now like can be precisely defined. Let's say like you want to design binder, now you can specify which basically tip atoms or like in the protein residues or like side chain residues will interact with the binder, like the target and the binder. So you can specify which atoms or like tip atoms or like some atoms that can interact with targets. And then also like you can for example, condition on active site. Let's say if you want to design an enzyme. So enzyme basically like it needs several catalytic residues. So you can basically really condition on this like catalytic residues positioning, and then you can try to scaffold these catalytic residues so that your catalytic residue basically will be geometrically correct after you generate the backbone. And you can also like condition on many different things, such as like H-bond conditioning, where you can specify hydrogen bond interaction between let's say like one chain of protein and then with the other chain of protein, specifying which one is the donor, which one is the acceptor. You can also like specify the rasa conditioning, which is like the exposure of the residues to the solvent. So you can specify whether you want this residue to be exposed to the solvent, meaning like it will be in the outside of the protein, or it will be buried inside the protein. So you can do like those kind of like specification. And all atom basically will provide more natural way to work with many small molecule ligands or DNA, because to design protein that can bind to this ligand or like DNA, you really need to specify like what kind of like interaction will accommodate this type of like binding. And the basically the pipeline in Baker Lab for the novel protein design, we always start with a function that we want. Let's say like you want to design a binder or like an enzyme, or let's say like a specific like protein interaction, or like DNA binder, or like small molecule binder. So we basically like specify that like what we want, like the function. For example, like we want to design a binder, so we have to find like what is the target of the for the binder, right? Like like your binder will bind to what protein, for example. And from there you can specify where your binder wants to bind, because let's say like you have like a target protein, and this is like in 3D. Let's say like you can bind to this side, let's say, or like bind to this side, right? So you specify it as your basically input. You specify the hotspots, and then you use RFdiffusion tree. Nowadays people use RFdiffusion tree most of the time, because that's the most developed and advanced diffusion model from Baker Lab. And you generate like basically thousands of structure. So in Baker Lab, usually we start to generate, let's say, 100,000 of backbones with different lengths. Let's say we specify the chain to be like, let's say, 150 to 200 amino acid in length. And then after that, we do like a pre-filtering before doing ProteinMPNN. So we just like remove backbones that have chain break, let's say, or like have clash between their secondary structure. I like those kind of like basically, like simple filter. And then after that, usually we put it into some sort of like ProteinMPNN version. Like, for example, if you work with ligand, there is like a ligand version of ProteinMPNN, which is called ligand-MPNN. There is also like a version of membrane protein MPNN, where the MPNN basically is like trained on different type of like proteins. So for ligand MPNN, usually like the MPNN is trained on proteins that are like soluble in aqueous solution, and also like trained on like proteins that have interaction with like small molecules or like other proteins. But for like, let's say like membrane protein MPNN, they train basically the MPNN with specific type of like proteins that are like like transmembrane in nature. So so it will have like more basically hydrophobic residues in those type of proteins. And then after that, after protein MPNN, usually like we generate around like 10 sequences per backbone. So you start with 100,000 backbones and you filter it, you might end up with, let's say, like 90% pass rate, let's say, because some of them, they might have like chain break and clash. And then you will end up with 90,000 backbones, and then you generate 10 sequences per backbone, let's say. And then after that, you fold, basically, try to predict the structure of this generated sequence from the MPNN using either like you can use RoseTTAFold or like AlphaFold tree. You you know like I usually use AlphaFold tree basically, and from there, after you try to predict, you will again filter because you want to only pick the proteins that matches with your design and your specifications of your inputs, right? So let's say like with binder, you try to predict with the target, and then whether after you predict your protein with your target, whether they show like the correct binding or like they just like randomly sit somewhere near the protein. So that's kind of like filtering, and then again like you filter for like chain break, clash, um, and also like other like filtering parameters that you might want to include. And after that, let's say like you end up with, usually like we try, after like we filter, we try to keep the picks to be only like around 200 to 400 proteins, and then you are going after that for wet lab protein characterization. So let me know if you are still with me, or am I clear or like not? I mean, you can always
Yes, Kak.
okay, yeah, okay, then yeah, I'll continue. So in the lab, usually like we do like in the form of like 96 well plates. Let's say like you have 4 plates, you try to express your protein in E. coli. Basically, it's, like this is like the time frame of the process. And then after you express your protein, you do purification with, like some kind of like purification tags, can be like, his tag, strap tag, snap tag, like different tags. I mean, you can choose based on your proteins. And we purify using HPLC, so that after you use like purification tag, you again like purify using HPLC to just get the correct fraction of the protein with like a single peak. Because oftentimes when you express protein, let's say like they might have like two kind of like form. One is the monomer, and then one is just the aggregates or like the dimer. So you want to basically separate between these two form, and then maybe test only the monomer. And then after that, you end up with your protein, and then you are ready to test for functionality. Let's say like if you design a binder, you can test it with SPR or like biolayer interferometry, or like for enzyme, you can test it with plate reader to see the activity. You design a substrate that can, like when upon like basically catalysis, there will be like some kind of like fluorescence. Then you can use plate reader. And again, these are like high throughput because you are testing around like hundreds of protein. So if you have like hundreds of protein design, like backbones or like, like proteins, and you test it one by one, you can imagine if you have 400, you need like 400 days, right? Let's say like one day or like, let's say one day you can test like 3 proteins. You will need like around like 100 to 200 days basically to finish all the work. But with this high throughput, you can screen it, like within less than a week, basically. Yeah, so that's basically like roughly the process for the Novo protein design in the lab. But obviously, like this is like like simplified, right? Again, like if you have problems, you have to debug your experiment. Let's say your protein don't express, then again you have to debug like why it doesn't express, like what's the problem, right? Maybe repeat the experiment, those kind of things. And now, like I want to touch some applications of the Novo protein design. So this just like recently published, like from the lab as well. I'm not part of this publication, But like basically we show like we can now like basically leverage protein design for many applications. For example, for antivirals, we have designed different types of like binder that can bind to this specific target epitope. We also like basically design a binder that can neutralize this, like toxins. For example, like from venom snake, where it binds like very tightly, like sub nanomolar affinity, basically. Or anti-cancer, where you can bind to specific receptor that will basically help to minimize the effect of this cancer growth. Or work with the immune system, where you can design a binder that can facilitate binding between these two uh, immune receptors that will basically have effect to the immune effects. The next one is enzymes, as I mentioned before. Since we can condition on this, like atom level, you can really specify what type of like chemical reaction that you want. For example, hydrolase or ligase, for example, like that. And then we usually borrow like active sites from nature. So let's say like we find like an existing enzyme, and then we take like only like a very small number of like residues that are involved in the catalytic activity. For example, like this one here, this is like, let's say like 3 histidine and then some substrate, and then you try to scaffold these active sites with the Novo backbones. And then after that, you can basically characterize whether your enzymes are active or not. And yeah, we basically the lab has published several types of enzymes, which like if you're interested in, I can share like some of those papers. Again, this is not like my work, but I'm just like introducing about the capability of this, like the Novo protein design. And now, like, I want to briefly touch to what I have done during my PhD, and also like what I'm currently doing in Baker Lab. I hope we still have time. Is it fine, or like I can just
Of
stop here?
course.
Okay,
We got
thank you.
time.
Yeah.
Yes.
Um, yeah, during my PhD, I basically developed this platform, which we called Maestro, which stands for like multi-axial entropic spring tweezer along around DNA origami. So this is DNA origami, no protein design here during my PhD. And the goal is like, I want to apply tension to a single molecule from multiple direction. So here you can see this is the DNA origami, the red circle, and then I can attach single-stranded DNA from this inner sidewall of the origami. And then this single-stranded DNA can basically connect to, to the single molecule of interest, and then it will basically apply tension to the molecule. So you can imagine this like, let's say if you have like a rubber band, right? So if rubber band just sitting on the table, there will be no tension, right? And let's say like if you hold the rubber band in between your two hands, right? So there will be like basically tension along the rubber band as basically like as your hands move further away, the tension becomes basically like higher along the rubber band. So similarly, because DNA is a polymer, you can think of this as an analogy. Then the tension generated by this single-stranded DNA will be dependent on the length of this diameter of DNA origami, and also like the contour length of the single-stranded DNA. And the molecule that I studied basically holiday junction. This is like a four-way junction that is basically very crucial during double strand break repair, or like homologous recombination. And I tried to study this dynamics under multi-axial tension. The way I did it is like I place a fret pair here, Psi3 and Psi5. So these are like fluorophores. So this basically like can be excited by a light, and then it will emit a different wavelength. And like this is like fluorescence resonance energy transfer. So Psi3 is the donor and then Psi5 is the acceptor. So when these two fluorophores, they're like close to each other, so there will be like energy transfer basically from the donor to the acceptor. But if they're like further apart, the energy transfer will be less. And then you can tell basically like this distance by monitoring the intensity between these two fluorophores under light microscopy. And this is just to show like the origami structure that we folded, which is like basically exactly agrees with what we designed. And I just want to show here, like, this is what we did basically, like multi-axial force. We basically like pull the Holly junction from multiple direction with different force, and we observe like different kind of like dynamics. The detail might not be like so important, but we show that Holly junction can interconvert between different type of dynamics. So this is what I meant before by observing the basically intensity of these two fluorophores. We can extract the energy transfer between these two fluorophores in different conformations. So you can see like different color indicates they're like basically in a different kinetic classes. The second project that I did was like about DNA transmembrane sensor. So the idea was like whether we can mimic this single pass integral protein with only DNA synthetic DNA system. So no protein, no amino acid, just DNA with some kind of like modification. So again, like I designed this simple duplex with two overhangs here, two toe holes, and then to facilitate the insertion of this DNA into the membrane, we put tags on this stem, this duplex stem with cholesterol molecules. So these are like cholesterol. So cholesterol is hydrophobic. So this cholesterol basically will facilitate the anchoring of this DNA through the membrane. And like we need this cholesterol because DNA is hydrophilic. So DNA will not spontaneously basically span the membrane without this type of like hydrophobic molecules. And we characterize with different assays basically, where we use synthetic vesicles, lipid vesicles, and then we show that we can detect basically we want to use this as a sensor, nucleic acid sensor, so we can detect a nucleic acid sensor inside the vesicles with the sensor through this confocal microscopy. The third project is all about basically engineering using DNA origami to store information and use super resolution imaging to read out the information. So the big picture is like, let's say we want to secure. This is like an alternative method to store data. So right now, basically, we use this silicon-based data storage, right, which requires a lot of like power, and silicon is expensive. But with DNA, you can just store it, like at room temperature, and then it will be stable, like for billions of years. So yeah, that's basically the motivation. And then we try to encode the information by creating a unique pattern on DNA origami. So DNA origami is the platform. Let's say we have like this DNA origami here, the square, and then we can just pattern unique, like arrangement of this molecules. And then we try to read the pattern by using super resolution imaging. As you can see here, the scale bar is like 10 nanometers. So this is way beyond the diffraction limit, or like the normal microscope can image. So like, for example, like light microscope is basically limited by the diffraction limit of the light itself, which is around like lambda divided by 2. Let's say we use like visible wavelength on like 500 nanometer. So the smallest distance that we can image with a normal light microscopy is basically around 200 to 300 nanometer. But with super resolution technique, we can image down to 10 nanometer or even like lower. Yeah. And in Baker Lab, what I'm doing is like we're trying to build a de novo protein. So maybe some of you are like familiar with this type of protein motors, triple A plus ATPase. So some examples of these protein motors, they are like helicase, or like this is like to unwind DNA basically, or clip X to basically um, uh, unfold proteins. So these two basically protein motors, they're like very important in DNA sequencing, as protein sequencing, because they help to thread the DNA or like protein into the nanopore, so that the nanopore can basically read the sequence of DNA or like the protein through electronic readout. And I tried basically like our idea is like we want to build the novel protein with a different fuel. So at triple A plus ATPase, it uses ATP as an energy sources. And this ATP basically will be hydrolyzed into ADP plus phosphate. But in my project, basically we don't use ATP. We use a different type of like fuel, which is a synthetic fuel. Um, like which consists of like these two dyes, and then basically we use this active sites thiozyme, where we have like three histidine, one tyrosine here, and then one glutamate. And we try basically, we borrow this from nature, from astaxanthin family, and then I symmetrize this using, like Pymol, and then using RFD3 symmetry to generate this C6 backbone. And then when we generate sequence, and then basically like it matches the AlphaFold 3 prediction matches with what we design. And then this is like the gallery of the novel protein backbones that we generated. And then now I'm basically like I'm in the process of like characterizing it in the wet lab. So this is like the idea. Basically, like we want to make conformational change when the substrate binds, and then after the hydrolysis of the substrate, basically the protein can basically change into the second state, and then it will basically like be the uh, the foundation of the motors. I think with that, yeah, I'm done here. So sorry, like it's almost 1 hour, but thank you so much for your time, and thank you for inviting. Yeah, I'll be happy to like answer questions and discuss with you. Yeah.
Wow, thank you so much, Kak. It was a really great presentation. I think if I want to ask something, it could be
Mhm.
for hours, but
Yeah.
sadly, maybe we have to stick to what related to our project. But
Yeah, and then we can always like basically you can
yeah,
always
I'll be
ask me
happy to
after that and
connect
then
with you.
discuss. Yeah. Yeah.
Oh, yes, thank you. Um, but before that, I think I forgot to introduce part of our team
Hmm.
from this item. So on today's meeting, I got my iGEMS team, which in this case the leader is Ferlin. Maybe Ferlin, you can introduce yourself.
Hello, hello, Ka. So my name, sorry, my camera is cannot functioning right now because of my signal. I'm so sorry. Okay, so my name is Farlin, and I'm here as the student leader for IJAM Brawijaya team with Shalfani.
Hmm.
Hello.
Yes, the yeah, Shell. You may introduce yourself.
Okay, thank you. Hello, Kak. My name is Shalvani that I emailed you before, and nice
Yeah,
to
yeah,
meet
yeah.
you. Thank you so much for the lecture today.
Yeah, yeah.
Yes.
Thank you. Yeah. Thank you for inviting me. Yeah.
And also we got two department that conduct the research, which is divided into dry lab and wet lab.
Hmm.
Maybe I will not. present all of the members, but we got some here like Kak Erman. He is one of my seniors in Brawijaya, and he also helped me with the wet lab. And also we got, oh yeah, and also this team is so diverse because it consists of many faculties.
Mhm.
We want this team to be a multidisciplinary one. So we got two Philcom guy here. Philcom is computer faculties, computer science,
Mm.
yes, so, some
Okay.
sort of like that. We got Dion and Nafis here.
Mhm.
Yeah, that's basically about it. And also because actually a bit introduction, this iGEM team is part of SynBio, which is stand for synthetic biology. It is a bit small to medium communities in University of Brawijaya
Okay.
that um, we gather people that like to research about synthetic biology and all of and also bioinformatics stuff. So I also invited some of them, which
Mhm.
from R&D department from our community.
Mhm.
Okay, so I think
So if I
yes,
can ask a question, like you guys are like all undergrads or like
yes, all undergrads.
okay.
IGEM is part of the Synbio, and I am the president of the Synbio right now on this year.
Mhm.
So yeah, this is basically
Okay.
what we're doing.
Okay.
We do a lot of research stuff on the environment, on the vaccine, on the many fields. So we gather people that like to do it.
Mhm.
I think I'd like to present our materials and also our project.
Mhm.
And I already listed some of the question I want to ask.
Yeah.
But I think while I'm preparing my slides, I want to ask Kak,
Mhm.
how did you find yourself pivoting into um, biology? Because you're a trained physicist, right?
Yeah.
Yeah,
That's
like
a good question.
yeah, because from what I read, a lot of
Uh-huh.
historical figures like Edwin Schrodinger's, Walter Gilbert, Francis Crick, all of those guy are physicists turned into biologists.
Uh-huh.
Is that
Yeah,
some?
yeah.
Is that like certain of time in your physicist life that you have to somehow apply your physics knowledge to something, or
Uh
what
yeah,
was the
actually,
thought? Yeah.
yeah, yeah. Thank you for the question. I think that's a really great question because I mean like um yeah, like my background was not like purely like or like pure like biology or like molecular biology, right? So but um what makes me want to pivot to like something more like uh bio basically biotech, like molecular biology, because I think during my undergrad well, honestly, like I didn't like biology during my high school because it was all about memorization and roting, right? So it's just like so painful, basically, like learning biology like that way. Although like I was interested in some part of especially like when trying to learn about like mechanism or like something like that. So, but everything in high school was like basically memorization and that's why like I focus on like more like something that I can really handle, like math and physics. But then after I did my basically internship and master in material science, I started to realize that um, um, in physics, I understand that I'm not like that smart to be a theorist, right? To be like a theorist in physics, you have to be like really smart or like genius. But I'm not that type of like person, basically. Like I'm not like really smart, but I'm just like curious, basically. And I attended like many seminars about like like multidisciplinary field during my master, about combining material science technique to study biology or like to study like molecules, like biomolecules. So that really excites me basically, like, and then that's why like I kind of like slowly pivot to biophysics, right? So I mean still physics, but like it has like biology flavor in it. And then during my PhD, I read many papers basically like, okay, understanding like how to create nanostructures with DNA is not enough because protein is the machinery. I mean, we need I need to basically like master or like learn about protein design from scratch, so that I can basically later in the future, let's say if I want to be an independent investigator, like be a professor or like whatever, like scientist, I can combine these two fields so that I can get a grasp on these two basically sides of like major biomolecules, and then maybe like from there utilize it for more like like therapeutics. Basically, like my goal is like I want to reach like like therapeutics, diagnostic,
Mm.
but I think I'm trying slowly to basically move towards. I mean, like trying closer to that goal, basically. Yeah. So yeah, nothing. I mean, like spatial is more like like contemplation within myself, like, and also like physics is already like kind of like saturated, and also like maybe that end for particle physics, like standard model has been described, and then it's basically agree with many experiments at CERN. So I don't know, like I don't see like much more like like future. Like I mean, maybe I was wrong if physicists is here, like they're like physicists here, but I just don't see myself like doing like theory and then I can become like successful basically. Yeah, because I'm not that smart. Yeah, so that's why like I prefer to a more basically like, and also like it's more fulfilling because I can relate it to myself. Like, okay, biological system, like we are all biological system, right? So if I can make something that can be useful for like everyone, like it would be like also like fulfilling doing this science for that. Yeah, yeah,
Yeah. Yeah, but when I saw about lab stuff, so I
uh-huh.
was amazed. Like you also know many terms. So I think
Yeah.
pretty multidisciplinary project, right?
Mhm. Yeah, it's just like learning from your project basically, and reading, right? Because I mean after undergrad your learning basically comes, I mean like you have to basically like put yourself like with your own basically like deadlines, right? Like I mean, okay, I want to understand this, right? I mean I need to basically like read and then like discuss with people. So yeah, I mean in undergrad obviously you take classes, right? And then that will give you
Yeah.
the foundation of the field. But I think during PhD it's not like the class is not as like intensive as undergrad, so you have to rely on yourself to kind of like catch up. Where and then I personally like like I still don't know many things, and yeah, that's why like I think learning is important. Yeah.
Thank you. I think we can proceed to our presentation. Is that
Yes,
good?
please go ahead. Yeah, yeah,
Yes.
yeah.
Um,
I
got
I
our
just need
slide.
to.
Oh yeah. Okay. We will wait for Kak Bima.
Okay. Yeah.
All right. Can you see my screen?
Mhm. Yeah, I can
Or
see your screen.
I think, but I think the Gmeet screen is blocking the screen. Can you see the Gmeet screen?
Yeah, yeah. The
Wait,
Gmit screen is basically right at the center.
wait, hold on. All right. Can you see it now?
Um, yeah, I can see it, but it's not like a full
Okay.
screen.
Is it right now?
Okay, yeah, yeah. Now it's good.
Okay. Wait. Oops. So, um, again, thank you so much, Kak, for
Mm-hmm.
gracing us today. Um, the on the slides, basically, I will tell you what are we and what we're doing right now in the lab, and also ask you some of the pipeline that we use, because somehow we have a similar pipeline, although maybe the application is slightly different, but we can discuss it later. So
Mm-hmm.
we are an IGEM team, IGEM, which stands for International Genetically Engineered Machine. Um, it is competition actually started as a community back in MIT on 2004, if I'm not mistaken.
Mm-hmm.
This community is maybe just like our community at UB. Maybe this community is for people that like synthetic biology, but it has grow until this year's, where it already grow as a competition where people can register and also compete in this iGEM competition. So what is an iGEM competition? It is an annual international synthetic biology event where campuses around the world can sign up, and they will be provided with synthetic biology tools and kits. Some of them are DNA, maybe in a form of plasmid and many things. So they have to come up with an idea how to utilize this synthetic DNA tools, to be able help community around them. So it is basically like a robotic competition, but the core is not maybe in robotics electronic stuff, but the core is synthetic DNA here,
Mm-hmm.
because they want to push the utilization of synthetic DNA in every day, on a daily basis around us.
Mm-hmm.
So yeah, it has been 22 years up until 2025, so it must be 23 years now, and it has grown significantly, exponentially, and right now it has 4,600 participants on the last years, and many campuses call it Ivy League, maybe, and even maybe campuses from Asia right now, from Indonesia, has joined this. Even I think ITB also has joined this since the first day. If you, I don't know if you have heard about this while you're in
Mm-hmm. No.
the, but
No,
yeah.
no, I've never heard that.
But I know some guy from ITB that firstly initialized the iGEM from ITB. I don't know.
Okay.
Yes,
Okay. Okay.
this is basically about iGEM, and this is our track record in iGEM from Brawijaya. Uh, we've joined this competition for 3 times basically, because the last time didn't count because it is a regional iGEM competition. It's not an international one.
Mm-hmm.
It is like a pilot project from iGEM, and it already discontinued. But
Mm-hmm.
the first three, we won bronze on 2014. We won gold medal in 2014. Had a hiatus until 2021. We won a silver medal again, and had a hiatus again until
Mm-hmm.
this year's. We want to continue this iGEM team in
Mm-hmm.
Brawijaya. So the timeline basically is, it is a 1 year project. It's not it's a long march of competition, and the final is on this grand jamboree event on November 13 until 16th. On this year's is at Paris Convention Center. So IGEM right now already moved their headquarters in on at Paris right now. Before it was at Boston.
Mm-hmm.
So a bit about the research. So the minimum entry to enter this competition is really expensive, to be honest.
Mm-hmm.
For registration itself, I think it costs you around 150 million rupiah.
Ooh.
Um,
Okay.
but on the as the offers, they offering us sponsorship. In this case, we can get a free DNA synthesized
Mm-hmm.
uh, up to 90 kilobase pairs
Mm-hmm.
accumulatively, and
Mm.
we also have fully control for BioBricks. It is like a platform to store any DNA sequence, maybe promoter sequence, maybe RBS sequence, plasmid, etc. So we get fully access to that. So we can um, ask for them to send all of those sequence as many as we like and anytime that we like on this IGEM period. So that's the benefit for us. So with this benefit, we have to work it all up in our lab and present the results on November. So that's basically about it about the
Mm-hmm.
IGEM project. Yes, I think we can skip this.
And about the presentation, is it like poster or like oral or like?
Yes. All right. So for the IGEM itself, because it is a long march of competition, we have
Mm-hmm.
to submit, I think, around 17, if I'm not mistaken. Maybe Fadlan can correct me. 17 deliverables, they call it deliverables. We have to present in a form of slides and video about our presentation, just like regular presentation. We also have to submit many little things such as safety protocols. And
Mm.
also one of the aspect that really important here is the human practices. We will discuss it later in the back of the slides. But on this human practices program, the IGEM like that everything around the world apply this synthetic biology field into their society. So they want to know what impact do we deliver to society around us. So we have to present that also as well, like what we did to the society. Maybe like workshop,
Okay.
maybe like this knowledge transfers
Uh,
with you. Uh,
oh, basically outreach, right?
yes, outreach.
Okay.
Yes.
Okay. Okay.
So yes, that's about it about the IGEM, and I think I want to present right now about our idea and project.
Mm-hmm.
So basically, our project started because our main PI is an MD, a medical doctor, and our second PI is Pak Tunjung. I think you know
Oh, I
him
see.
very well. He
Yeah,
mentioned
yeah,
a
yeah. I know
lot.
him.
Yeah,
Oh, okay. So okay.
yes. Those are two of our primary PI, and because our primary PI is a medical doctor, she come up with problem that are happening in Malang and especially Indonesia, which is MASLD.
Mm-hmm.
But I don't think we will discuss it more further because it's just like a study case for us in this project. But the thought process is started from this disease, muscle D, which is basically when your liver has a lot of lipid and it disrupt the function of your organ, and it can lead up even to cirrhosis, which is a reversible condition. But right now, this condition is somehow underdiagnosed. Like many people already know that they've been diagnosed with muscle D after they already come to a stage on the cirrhosis stage where it is already irreversible. Because on this stage, um, people can know the symptoms because the symptoms is can be appeared.
Mm-hmm.
You can see your skin turn into yellow, your eyes turn into yellow, but before that, there's no signal, there's no symptoms. But it is sad, right? Because if you already have a cirrhosis, it is already irreversible.
Mm-hmm.
Mm, Started from that idea, we think what a new diagnosis method can be applied to early diagnose this stuff. So one of the method that came up to us was the usage of metagenomics method,
Mhm.
which is basically a method where you sequence a sample of DNA in samples, but the samples does not have to be specific. It can be um, consist of many DNA. For example, you can extract that DNA from a soil and just ask for the machine, which is NGS, and in this case, to read any DNA of it, any
Mhm.
DNA that happen to be on the soil sample, and we can somehow analyze it. What are the microbial communities on this soil sample? And we can get this data like the relative abundance, for example, maybe on the soil, how many percentage that this bacteria has on it, how many percentage this another species has on it. That's basically the
Mhm.
metagenomics method. With this, we thought that, oh, maybe there is a biomarker that represents certain diseases, and it is also really emerging research right now, because many scientists right now utilize this to able to decrypt what biomarker is responsible to certain disease. Right now, there are many scientists use this pipeline maybe to diagnose even like um, dementia, maybe Parkinson, even immune systems disease, any of that, based on guts, because
Okay.
somehow our guts represent
Mhm.
profile of our guts represent what the status of our organs.
Mhm.
For example, like this, on the cirrhosis patients, you can see there is a significant increase of Firmicutes bacteria.
Mhm.
That's basically about it.
Mhm.
And why it is so important? Because our guts, yes, it is a microcosmos of its own. So it's somehow connected with liver, kidney,
Mhm.
bone. Even right now in Indonesia, if you don't know, we can sequence our gut that to know the status of our skin for skincare purposes. Uh,
Mm. Okay.
that's
Okay.
emerging right now. So, but on this pipeline, it is first expensive and also not accessible to many Indonesian, right? So we think, how can we provide point of care test, which is a test biosensor that is practice, that is practical, that is accessible and also cheap, and can detect some of the biomarkers that responsible to any of uh, increase in gut microbiome. So it is basically a bacter sensors. We create a bacteria that can detect other bacteria. That's basically the main
Mhm.
idea. So because we want to create pipeline to detect other bacteria, we think what detection machine that we want to imply on our project. And turns out we came up with nanobody.
Mhm.
Why we use nanobody specifically? Because at first we thought that, oh, the easiest one is to just produce an antibody and let it, you know, let it dock, or let it yes, let it dock to other proteins. So we, if it is docked, then we can imply that that is a positive result. But turns out the antibody is really big. It is 10 times bigger than the nanobody. And from what I know, the bottleneck of synthetic biology is always the expression of the protein, because we can design a really good protein, but when we express it, maybe it did misfold, it did degrade many things. So one of the novelty here, why would not we use new method, which is right now using nanobodies, which is is more compact antibody, and we design it ourselves. However, um, this nanobody is somehow really hard to design. It's really hard to design a good binder one, because you have to, just like in classical immunity extraction, you have to immunize the um, maybe rat. In this case, nanobody is derived from camel.
Mm-hmm.
You have to humanize the camel, and then extract the lymphocyte from the camel, and etc., etc. It is labor intensive, and also, yeah, time consuming. So I came up with another idea. How can we utilize something that can bypass the system?
Mm-hmm.
Because I'm interested in the bioinformatics. I read and also listened to one of the presentation from David Baker's on CASP lab, and he introduced me to the Novo design. So combining those idea, the red line is we want to create a nanobody that's specific to other bacteria protein surface.
Mm-hmm.
It can detect other bacteria,
Mm-hmm.
and express it in E. coli. So we
Mm-hmm.
can create this agglutination effect, where if the result is positive, then you can know directly from your naked eye, because our goal is to create a point of care testing, which has to be accessible and also cheap.
Mm-hmm.
That's our thought process about our project. And this is our stage. We already um, did some research about the biomarkers. On this research, we use MASLD as our study case. Because of that, we use one of the biomarkers, which is the increasing of Anthracoccus faecalis. But to be honest with you, this iGen project is never meant to be um, producing maybe actual research that you can publish. It's never meant to be like that. It's just how your team can do a proof of concept of
Mm-hmm.
what your idea.
Okay.
So our our main goal here is to able to provide proof of concept. So it's like, for example, if you want to, yeah, maybe create a cancer drug, it never meant to be cure the cancer, but just just the basic proof of concept, because the timeline is really short for
Yeah.
the web lab stuff. Yeah, this is our pipeline, and then this is also I brought up from the Baker's Lab presentation. This is the
Yeah. I mean,
dry.
I also use Baker Labs basically.
Yes.
It looks very similar.
Yes. Um,
So yeah, yeah. But before that, like, so this is like what, 10 months, basically timeline, right? And
yes,
then
10 months.
and how do you um distribute between like wet lab and dry lab
Right.
basically?
Yes. So the dry lab here will basically predict and also run the simulation of the verification of each de novo designs, and the wet lab will express it on E. coli,
Mm-hmm.
which happen to be, we want to express it in the surface of the E. coli. Because of that, we um, diffuse our nanobody with beta intimin, which is the anchor protein in the transmembrane of our E. coli. Right now, the design of nanobody is already done, but I
And
think
how
I
big?
want to
How many?
criticize.
Yeah, if I can ask,
Yes,
like
yes.
like how many design you start with,
All
and
right,
then
all right.
like how big is the,
Yes,
because,
that's
well,
basically what I want
again
to
I
ask. Yes,
yeah I've never worked with antibody, right, or nanobodies,
yes.
but I think with antibodies there are like specific kind of like,
Yeah.
with antibodies. I mean, there are like specific like S, like VHH, or like like
Yes,
VH,
yes,
VL, like SCF. I mean, like like fragment, right?
yes,
So those are like classification, right? I don't know, like
yes.
with nanobodies whether you have that,
Um,
because I think Baker Lab also like developed specific RF diffusion, but I think it's based on RF diffusion one,
yes,
where
yes,
they mainly train the model based on the antibody structures in PDB, right? So, and then they try to basically design the CDR domain, I think. Um, but I'm not sure. Like, yeah,
yep.
I want to know more about what you, yeah.
All right. I think we can deep dive right now into the nanobody design. So nanobody was first extracted from camel.
Mm-hmm,
It was firstly discovered that camel has a different type of antibody, not like any other mammals. Turns out their antibody system, their antibody structure only consists of one single heavy chain.
mm-hmm,
So their recognition site, other mammals have two, right? Um, Light chain and heavy chain
mm-hmm,
on the recognition side, but on this camel, they only have one single chain that recognize antigen. And like you said, what group it what what group, I think it is same as single heavy chain VHH.
mm-hmm,
The nanobody terms is just another term of that because it is small, people call it nanobody, but I think the more scientific name is VHH, if I'm not
mm-hmm.
mistaken.
Yeah, yeah.
And to deep dive about our design, um, we did design with the antibody pipeline. So
Mm-hmm,
from what I know, correct me if I'm wrong. Um, I think Baker's Lab developed a lot of pipeline, right? Like
mm-hmm,
they tried with maybe peptides, RF peptide, if I'm not mistaken, how you can de novo design peptides. They tried with RF antibody as well, where you can design antibody or antibody with this de novo approach. I think the difference from what I read is just they fine tuned each model to each application.
yeah.
So because our application here is to design nanobody and yeah, nanobody, we use the RF antibody pipeline,
Mm-hmm,
which consists basically just the same, but the paper says that it already fine tuned into the
yeah.
uh, application of nanobody. That's what we use. Um,
Yeah, yeah, I
I
think you're right. But yeah, I think you're right. Yeah, it was based on like RFvision one, right? So
yes, yes, yes,
yeah, I can check again with like some of my colleagues, like whether they have like, I mean, again, like you have design, but like it's
yes.
just like for my curiosity, and also like maybe like for your information later, if they have like basically developed the same thing with the RF diffusion 3. But anyway, I think you're
Mm,
right. Like, yeah, I mean, I think they just published this year, but I think the preprint has been out since maybe 1.5 years ago. Like
yes,
this
yes,
antibodies, basically.
yes.
Okay, okay, okay.
The preprint already.
Yeah, 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 yeah.
At first, I was thinking, yes, there is new uploaded RF Division 3, right?
Uh-huh.
And it says it is more accurate and etc. etc. But I don't know if should I choose running each simulation separately with each new models, or I should just use a model that somebody has fine tuned before. Uh,
I
we
think,
we chose that because I think they already consider it
yeah.
to be able to fine tune it, right?
Hmm. Yeah. I think, yeah, I mean, what you have done there is like, I think completely, like justified and right? Because, like, it's published and they show like, okay, it's fine-tuned, as you said, and
Mm.
basically specialized in antibody. But let's say, like, I don't know, like, in terms of like time that you have, right? Let's say if you want to make more, maybe you can try to do RFdiffusion 3, but I'm not really sure whether there is like specialized RFdiffusion 3 that is trained specifically on
Yeah.
those subset of like antibodies and nanobodies basically. But 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 I don't, like, from my understanding, like,
Mm.
whether you real like whether just design binders that can bind to this two are enough, or like it has to be like within the class of basically antibodies or like nanobodies, right? I don't know, like, I mean, that's just my question basically, because designing binder, I mean, it doesn't have to have basically a unique, like conventional basically shape, right? But of like antibodies or like nanobodies, that's like, like
Mm.
um backbones, like with like alpha helices, beta strand, let's say, that can basically like calm this into aggregation, right? Like between the bacteria, like between the bacteria, and then facilitate basically this agglutination. But I think, yeah, I mean, because you have done this, and then I think it's it's justified. Yeah, yeah, should be fine.
All right.
Yeah.
So for the design that we produce, we design nanobody for each target, which is this protein, EBPC, this protein, and also this protein. We
Oh,
utilize three protein targets on the surface of Anthracoccus faecalis that has been proven to be conserved in faecalis.
okay.
And then,
Um,
hmm?
so you design three different binders basically, like three different
Yes,
nanobodies.
basically we designed nine.
Oh, okay.
Each protein, and each protein have three different hotspot
Okay.
positions.
Okay.
But yeah, and from what I read on the RF antibody paper, they said that what lacks on the model is the filtering pipeline.
Mm-hmm.
They honestly say that many uh, model didn't translate into the wet lab because of the filtering pipeline. So here I came up with a bit of a novelty where other than filtering from the structural filters from the RF RoseTTAFold data, we
Uh-huh.
also filter it from the predicted binding energy, and also we did some molecular dynamics simulation to able to proceed only the nanobody design.
Okay.
We have done all of the designs, and this is basically our filter pipeline. There are PAE,
Mm-hmm.
align RMSD, LDDT as well, and
Mm-hmm.
also the RMSD of the CDR region, because for the context on the
Mm-hmm.
nanobody structures, what
Mm-hmm.
we mutate here, or what we predict here, is only the CDR region. All of the nanobody, we use the framework which is
Mm.
called,
I see.
uh, I don't know the exact name. It is so hard to pronounce, but we utilize this framework that considered as plastic framework. So
Mm.
research say that if we mutate this CDR,
Mm-hmm.
it does not affect the compactness of all the structures. So
Okay.
we only mutate this three regions on our designs.
Basically, you design those three regions using RFD, right?
Yes,
Okay. Okay.
because it is the sequence that directly dock into the antigen target.
Mm-hmm.
Um, on the MD simulation, which we still on progress because we still use only 10 and we haven't done it to all of the designs, but this is basically what we did for our designs. This is one of the top designs
So
for
from
the
MD, what kind of like information you will
yeah,
use basically?
we will use the RMSD of the residue, sorry, of the backbone uh, to check whether it is has it did fluctuate much or not, and also we uh, uh, search for the hydrogen bond, how many hydrogen bond
Mm,
did occurs on the
okay.
simulation, and also wait, I think I haven't typed the real one. Number of hydrogen bonds,
Mm-hmm.
and also the radius of gyration. That,
Okay. Of
uh,
the protein
yeah.
of the
Yes,
what?
of the nanobody.
Okay. Okay. Okay.
Yes, that's basically about it. In this MD, I think we still learn about it. I think if you have insight for it, it will be perfect because we are also new into running simulation on nanobody and target complex, and we also run with the top best. For example, this one is for the ACE protein target. This is the top one, and we yeah, there are many hydrogen bonds as we see on the slide, and yeah, they that is basically how we filter our nanobody design,
Mm-hmm. So
and we also
you
already
did you filter the design at the very end after MPNN, or like how, is it like only,
yeah,
is it
actually
like
we
one?
only screen it after the RoseTTAFold design
Okay.
has appeared. But I listened to your presentation and you said you did filter on the backbone, so
Yeah,
I haven't
I
heard about
mean,
it. Can you tell me about it?
yeah, it's just like sometimes, I mean, because like, I mean, again, the diffusion model is not perfect, right? Sometimes you have like chain break, right? So you obviously I mean, you don't want to continue with those type of like basically backbones, right? That has
Yes.
chain break, right? Otherwise, I mean, it will just like be a waste of compute basically, right? If you just continue with those
Yeah, yeah,
Yeah,
it really fastened up our
yeah,
prediction.
yeah. I mean, even like I only use A6000 sometimes, or like A4000. So
Oh,
like thing H100. Is it with H100, right?
yeah,
Yeah, it's
no,
already like
A100, A100.
A100. Oh, okay. What is the
Yes.
A100? Oh, yeah, yeah. Okay, 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 okay. Yeah, I think
Oh,
yeah, it's pretty good.
I
I
think it's
think.
pretty
Yeah,
decent.
yeah, yeah.
And we did so. Here it is the raw file of our designs.
Hmm.
So at first, we design only 1,000 designs.
Mm-hmm.
100, 100 backbones, and then times 10 with the protein MPNN sequence prediction for each design.
How
And
long
then
did it take to generate 100 backbones?
100 backbones, I think just 1 or 2 days. Is that right, Nafis? Two
Yeah,
days.
Oh,
it took
okay.
2 days.
Days. Yes. Is it
And
long?
mm, 100 backbones 2 days. I think it's I think it's normal, but I think like if you can have access to the A100, like pretty often, you can generate, like even more basically. And then
Uh,
specify basically like play with the parameters. I don't know like what kind of like parameters, like basically like like now length, let's say,
yes, yes,
and
yes. That's
and and
also what
and
I wanna ask.
uh-huh.
So because to be honest with you, with this new pipeline, I really don't know what parameter should I use. Is it like good to do this? Is it like restricted to do this? Because there are so many parameters. For examples, on this CDR designs, we can make the design predict in each of length of acid sequences that we want to add, but I don't know whether it is will make it the nanobody design better or not. So on the CDR length, we keep it default. But what do you think?
Yeah, that I guess like I think it's a very great question. I would say like because I don't have direct experience with antibody, right? I
Mm,
think it's important to try to play with this parameter again. Like you will not try, like a very big space, right? Like like but at least like try, like maybe like 5 different parameters so that just to get like like more like diversity
mm,
of the backbones, right? So with more diversity, maybe like you will increase your success rate after like doing several steps of filtering rather than just trying one parameters, right? So maybe like some of your sample, like many of your samples are like like similar, right? With similar length, but if you try like different parameters, let's say like like one parameters, most of them they fail, but you still have like other parameters that might end up like passing the filter. But I would say like yeah, with the length I have no experience really, honestly,
yes.
like how it should be like the CDR. But I would say like I don't like for your target protein, is there like already like an antibody that like natural antibody that can bind to that? Maybe you can try to look like on what natures
Mm,
have, like what kind of like like distill some like like some values from natures, right? Like try to get like whether this like ace protein or like other two proteins that you want to target, they already have like like antibody partner or
mm,
like binding partners that bind to specific epitopes that you want to target, and then see like how those natural protein CDR basically in terms of like the length and properties. So maybe like you can try to learn from that,
mm,
and then try to borrow like some like parameters, like what are the lengths, for example, and then you other
mm,
parameters basically like how they like how the hydrogen bond, let's say, like I don't know if you can condition the hydrogen, because I've never used this RF antibody pipeline, so I really don't know exactly how it looks like. I can take a look on that, like basically like from their GitHub maybe, and
Yes.
let you know like what I understand, or like maybe ask some people that work on it, because I think many of them they already left the lab, I guess, because
Oh
like
yeah.
this like
Oh,
yeah, but I can ask some of my colleagues maybe that are still working on antibody, and then try to get. But I think my suggestion is like if you can get pretty often compute access, just generate more backbone maybe. But yeah, again, in the end,
yeah.
the downstream will also like be expensive, right? You have to predict. I think MPNN is pretty cheap because it's just CPU.
Yeah, yeah.
Yeah,
The problem is
uh,
always with the backbone design. And
yeah, backbone design is it's not like expensive, but and not like very expensive, because like, but still like, I mean, that's I think the most like heavy compute part. And the prediction, I think it should be fine, I guess, because uh,
mm,
I don't know. Yeah, I've never used RoseTTAFold antibody specifically, so I don't know how fast it is basically. Yeah, yeah.
how many days it took, Nafis, for the Rosetta?
It took so the design, the backbone, it took 1 days, and the Rosetta Fold for the validating it took 1 days as well. So 2 days
Mm,
in total.
Yeah, yeah. I think 2 days is until all of the design is finished.
mm.
And because
But
of what you said, that maybe we lack of number of designs, we run the second prediction, producing 1600 designs. Right now we produce 200 backbones designs, because we follow what the RF antibody paper use as the parameters.
mhm. Yeah, I think they mentioned like the success rate is like around 2%. I think like up to 2%. I think I don't know like
From the paper.
so. Yeah, from the paper. I need to check the details, but
Yes.
if the success rate is basically like single digit, yeah, you can just kind of like do some kind of like simple calculation, right? I mean, if you want like let's say five at least,
That
like
means
in
two
the
in.
end, like how yeah. I
Yeah.
mean, that's just like simple back calculation, but again, like different cases will be different. Like I mean, like we'll have different success rate, right? Like some, I think some target they're like very hard to target.
Mm.
Some, and we don't know like about your target protein, because I don't think like anyone has done it, or like I don't know like like
Yeah,
whether
I don't think anyone has done it
yeah.
specifically.
So that's why like yeah,
Yeah.
it's really like I think if you really have like access to compute power, yeah, just
Like how
play
many
with the
backbones design would you recommend? Like
um yeah. Well, honestly, in the Baker Lab, some people they started minimum maybe with 10,000.
the
Some of
backbones,
them,
or
yeah, the
after it
backbones,
replaced with
the backbones, just the
oh,
backbones. With, but again, like because we
it
have like
means
so much compute, we have so much compute
it
power,
means
right? I mean, some
10,000
people,
days.
some people they do like 100,000. I mentioned, right? So, but then, um, well, the question is whether it's necessary or not, right? In the end, like we want like a few working designs, basically, right?
Yep.
Right. So I think if you increase the number of backbones, I think the chance to get like some that will pass all your basically dry lab filter will be higher, and then maybe it will translate to higher chance in the wet lab. But again, even like you can do other methods. Let's say like you can always also like do hallucination, like cycling between. Let's say you have like your current backbone, right? Like
Yep.
1600, right? Let's say you can again like use that sequence design with MPNN, and then predict with RoseTTAFold, and then cycle it basically. Uh, and then because when you predict with
Mm,
RoseTTAFold, it will not be exactly as the same as what you design. Right? There will be like some change, right? Because And then as you basically cycle through these steps, you might get like a little bit like different backbones basically. I don't know whether that will be cheaper, but that that is kind of like one way basically cycling with
Uh,
MPNN and RoseTTAFold to get generate more backbones. Let's say like, or you can do um
like an iteration cycle. Right?
yeah. So that one way basically to generate more possibilities. Yeah, if that is like cheaper in terms of like compute power. Yeah. But that's just like basically like some ways. Yeah. You can do partial diffusion as well to generate more diversity. Basically like you have like let's say like few good designs that pass all the filters, and then you can try do partial diffusion, basically like diffusing a little bit so that it will still retain that secondary structure arrangement, but it is just like basically wiggle, like a little bit of the backbones, and then that might basically help with the optimization of the sequence that will fold into this that 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 backbone
Mhm.
basically, because you are trying to get out from that, like remember like when I show like local energy minima, right? So if your structure basically kind of like in that local energy minima, MPNN might have difficulty to get out from that local minima to get to the best sequence basically. So oftentimes in the lab as well, like when let's say like people after filtering, like the final filter, they will again like do another round of like, or like two rounds of like partial diffusion to get basically more just like possibilities of backbones, and then try MPNN with that. Yeah, yeah.
How about the number of MPNN design, Kak?
I think MPNN 10 is 5 to 10 is in, yeah, good enough. I think. Yeah.
Well, it doesn't significantly affect the designs.
Um, no, because well, again with MPNN you can play with the temperature as well, right?
Yep.
Uh,
Yep.
so if you put like like a bit higher temperature, you can get more diverse sequence, but still like
Hmm.
you don't know like whether those diverse sequence will fold correctly into your backbone. But I think I would say like try first with higher temperature, maybe like 10 to 20 sequence, and then fold those, like uh, uh or like do like two different temperature basically. Let's say like small, and then try a little bit higher, and then just like predict those sequences. Yeah, basically like don't stick with one parameter. Try at least like two or three parameters, something like that. Yeah.
I see.
Yeah. Because those like we don't know, right? I mean, those are like computational, right? I mean, like
Yep.
if we know like how it affects, and oftentimes like it doesn't translate from one problem to other problems. Yeah.
Of course.
Some design problem might get away with like low temperature, like sequence MPNN. Some problems might need higher temperature. I mean, I think, yeah, trying like at least like more than one thing, like will be beneficial basically.
Well, apart from the design as well, we also um, change some parameter with the protein target.
Mm.
So from like I just said, our target is three proteins that has been conservatively on the surface of faecalis. But my thought was, I think if I don't really understand about the designs, I can just keep it default, and maybe what I need to change is the hotspot residue of each protein.
Mm-hmm.
So, for example, on the one protein EBPC, take EBPC for example, we conduct three designs, each 1600 with different position. Like this one on the hotspot residue of 61 until 67. Um, we also tried again with this positions and this position.
Mm-hmm.
But the hotspot residue picking, I use how exposed the surface to the, yeah, how well exposed the surface is.
Mm-hmm.
So I choose three different locations because from the prediction, I think the prediction is called SASA,
Yeah,
soft line
yeah, yeah,
surface access, something like this.
yeah, yeah, yeah.
Um, I pick the most exposed one,
Mm.
so I can use it as our hotspot residue target. Is that
Yeah,
a
I think
correct?
yeah, I think that sounds good to me. I mean, like, and very very justified. I think because um that will be very easy basically for the antibodies to target, right?
Yes.
And then um you say like you pick like three, right? Basically, right?
Yeah, like my hypothesis was maybe on this side it's not well recognized, or maybe some misfolded happen with the protein, but on the other side, with
Mm.
the three chance, maybe the successful
Mhm.
rate will be much higher.
Okay, okay. Yeah, I think that sounds good to me. Like, I mean, yeah.
uh, because I don't really have the actual data that people already conducted the experiment, like they already know the catalytic side of this protein, so I just
Hmm.
use prediction to predict basically.
Yeah, I think using Sasa, uh, uh, I mean, like, um, how to say, it's it's basically like like one way to see whether
Mm,
those residues like exposed or like buried. So if it's buried, so it'll be like basically like very hard to target, right? But
yes.
exposed will be easy. Other than that, usually like people also like try to uh, maximize the hydrogen bond. Maybe like you can try to pick residues that can make hydrogen bonding. Like either like become a donor or like acceptor.
Yes.
Yeah, those type of like residues. Maybe like try to
Mm,
something like that. Yeah, 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 yeah. So that will basically like give you like I
yeah,
think if it can make like hydrogen bonding, the binder will be like I think like like I think it
basic.
will have the affinity basically of the binder. Yeah.
Yeah, it
Yeah.
also included on my calculation. Like I pick up residue that can act as a hydrogen acceptor.
Hmm. Yeah. Then yeah, you're doing the right thing. I think. Yeah.
But I think for you, the most significant one is the backbone design. Yeah.
Yeah. I mean, if you can get more,
So,
then yeah,
well,
just yeah.
how about the filtering and iteration cycle that you mentioned? Can we dig deeper into that? Maybe we can apply it into our dry lab,
Um.
because right now we just use as it is.
Yeah. I think using as it is already like because I mean the paper already show that it works for them, right?
Mhm.
And again, you can be more creative, like you did like with like MD, right? I don't have background in MD simulation, so I don't know much like how to run MD simulation. I don't know, like you use like Gromax or like what, like
Gromax, yeah,
yeah,
we
yeah, yeah.
use.
I mean, I know a little bit, but I've never run by myself. And yeah, in our lab, I don't think like people use MD as a
Mhm.
filter. Usually we use basically like that PAE mean PAE,
Mm,
and I think PAE is really important. I think you mentioned like less than 10, right? Um,
yeah,
Maybe if you
yeah.
can get it down even like smaller to less than 5 or like less than, because that will
Mm,
tell like how basically how well they are like basically to each other, right?
yep.
And PLDT you mentioned like bigger than 90, right? So you can also like add, let's say, the PLDT of the residues that are like basically making or like facilitating the context. Right now, like I don't know, like this PLDDT bigger than 90. Is that like for all residues or like specific residues? You can basically play with that. I would say like maybe like bigger than 90 for all residues, fine.
Yep, yep.
But let's say if you maybe want to add more filter, you can have PLDTT for specific residues that make that facilitate the context between the target protein and the nanobodies, and then you can maybe like bump it higher to 95, let's say. But in the end, right, you want to have like passing designs, right? You don't want to have
Yeah.
zero passers, right? So you will try to basically, like make a trade-off within those filter, right? Within your limitation, basically, right? I mean, let's say your limitation is like you cannot generate like so many backbones. Okay, fine. Then try to see like, or like let's say like you have like in the end 20 passing designs, and then you just list all those basically values like PLDDT, PAE mean PAE,
Yep.
um, and how many hydrogen bond, um, and what else? Yeah, those kind of things basically.
Yeah, the RMSD as well.
RMSD, yeah, I would say like it's good. Yeah, RMSD usually like
Mm,
we try below 2, right? Between design
below
and prediction basically. Yeah,
yeah, below two.
yeah. So yeah.
Well, for us, on our case, the harsh parameter was not the PLDDT.
Mm,
Maybe Dion or Nafis can share our CSV because I think it predicts good enough. So it produced a lot of PLDDT that pass below, not below, yeah, that passed the 90 scores.
mm-hmm,
But the harsh parameter was on binding energy.
mm,
So it is not recommended by the paper, but we
mm,
we added by ourselves. So we use Prodigy model to
mm.
predict the binding affinity of our complex. And
Okay, I see.
binding
Do
energy
they use this model as well, the Prodigy model in
on
the
the
paper?
paper,
Yeah.
I don't think so.
Okay.
Yeah, it's
Yeah.
just something
Okay.
we add up on the way.
Mm,
Yeah, this
yeah.
is one of the top designs. What protein is this, Dion?
This is the ACE protein.
The ACE protein.
Yeah.
And some of the protein targets does not produce as robust as the others. So some of them only pass at the end of the pipeline. They only have like three designs that pass the pipeline.
mm-hmm,
While some of the designs they have like maybe around 16 until 20 if I'm not
mm-hmm.
mistaken. So
Yeah.
is that count as a disadvantage or a flaw?
Well, again, no, like I mean, like I think it's good to be very careful, right? And in the end, like how much basically resources you have to test this, right?
Yes.
Right. So because I mean these metrics are good, but sometimes like like testing more. Let's say like even though like it maybe it's like in the how it's like like maybe it's just like barely like mid or like the delta G, right? Like maybe like
Yeah, the
minus
dosage.
9. You mentioned like minus 10, right? Maybe
Yeah,
minus
our
9.
passing line
Like
is -10.
yeah. Maybe it's just like barely made it. Like or like just slightly below. Maybe you can include them. Again, like it's basically this is like be- because you don't know yet, right? This is like you have never tested this, right? Because usually
Yeah,
like
on the
in
wet
our
lab. No.
lab, like in our lab usually like we do like first design campaign. Okay, we have like loose filter. And and then
I
after
see.
testing in the wet lab, we see like, okay, with loose filter, we only have like a few. In the next design campaign, we'll make it like more strict, basically. So again, like given like your constraint and limitation, and also like resources, I would say like, let's say like time is the constraint, right? Because you need to get something,
Yeah.
like for this by the end of this year. And if you have like more resource, maybe it's a good idea to, like let I say, like I said, generate more backbones, generate like let's use like more diverse backbone, and then like and see whether you can get higher passing number. If not, then maybe you can relax some filters a little bit, something like that. Because these filters might not linearly translate into success in experiment, right? So yeah, yeah. I mean, it helps basically, but I mean, no one knows until you test it, basically, right? Yeah,
Yeah.
yeah, yeah.
From what you've been doing there, like how many percentage usually you have?
So
Like,
right now, yeah, I mean, because I've just like started my project, right? So for me, like, I think I designed like 10,000, and then in the end, I end up with like around 300 sequences.
hmm,
And then I tested, I think last time I haven't tested all basically, but like I think few of them, like maybe 10. Yeah. So work in the wet lab. So yeah, that's my basically experience. But again,
hmm,
it depends from problem, like like from problem to problem, right? So I think binder design is, I think sh- I think binder, but not antibody, right? I mean, antibody is still like basically I think difficult. But binder, I think just binder, like normal binder, right? I think it should have like higher success rate. But I think antibody, especially like like dealing with CDR
yep,
specific, like target, might have like lower. So that's why like
hmm,
it's very hard to say. Yeah, yeah. But
like,
again, I think what you are right is like you're doing it right. It's just like like for your efficiency, right? If you have like, let's say like you say like you have three, but if you can accommodate more in your wet lab in
hmm,
terms of like like uh, experimental resource like money, etc., maybe you can test more design. Yeah, something like that.
Yeah, I
Yeah,
think the best practice would be to make as many backbones as possible, considering the success rate is single digit, as you
yeah,
mentioned.
yeah, yeah. Yeah,
And maybe I'm planning to do the filtering right after the RF diffusion.
yeah, you can add that basically and see like how much like as after that how much, because you filter there and then you again you have to filter at the end, right, basically.
Yeah,
Yeah,
so
so that you don't compute backbones with chain break, whatever, like it will just
yeah,
be a
that's
waste of
the
like
thing.
compute.
So I
Yeah,
don't
yeah,
we don't waste time on the
yeah,
Rosetta.
yeah. But again, if you don't have like chain break or clash, then it means like I mean the RF diffusion already doing a great job basically, so you don't have to worry about it. But it's just like a good check basically whether you have
how can
chain
you check the chain break?
chain break. Usually like it's just like the C alpha distance between the adjacent C alpha, like within like certain distance. I think it should be around, I forgot the number, 4.5 angstrom. Um, I can check again, but for the clash, I think it should be like higher than 3 angstrom. I think between the tip atom of each residues. Yeah, but those value you should check. Yeah, yeah, yeah.
Is there like a tool that you use for this, or like do you write the script by yourself?
I mean, we have basically like script, right? I mean, it's just like basically using BioPython just to check distance between, like neighboring C alpha, and then, if there are like atoms that are like too close to each other or like too far, it will basically show chain break, right? Yeah.
So you just use those two parameters for
Yeah,
filtering
it's
the.
just like initial filter basically, so that
uh,
yeah, 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 yeah.
what are the parameters again, Kak? CA.
Uh, C alpha basically like that chain break, and then clash also basically
Oh, C alpha is for
the
determining
chain
the
break.
chain
Yeah,
break.
the carbon alpha, right? The backbone basically.
So we have to calculate the C alpha distance. Am I right?
Yeah, yeah, yeah. But again, like
Okay.
if you, yeah, I mean, this is like you can basically ask, like Cloud or like ChatGPT to make the
Yeah.
script for you, right? So it's very easy. Or even like maybe Baker Lab in there, I don't know, like check basically
Do you use
the
Cloud, Kak, in your Baker Lab?
yeah, yeah. I mean, I think
It's
here
really good for biology.
we use Cloud. I mean, it basically expedite a lot of
Yes.
our work basically, right? I mean, you don't want to spend time. I mean, it's good to write your own code, but again, like the focus is like to get something that is working, right? Like I mean, like
Yep.
protein.
Yep.
Yeah, yeah. So, but again, like yeah, it's like
Can automate much.
yeah, 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 yeah.
So um to conclude, I think we have to increase the backbones and then the backbone numbers, and then we can initiate initial filters using C alpha distance
Yeah, yeah.
after the
Uh-huh.
RF diffusions.
Yeah.
And also I think MD is not really significant from your perspective as a
That's
filtering agent.
yeah. That's I've never used MD to filter, so I don't know like because in MD you might get the trajectory, right? Whether the protein will go crazy or not. But based on my experience, if if it predicts well using RoseTTAFold or like AlphaFold, usually it will just be fine. Yeah. So and for a number of hydrogen bond, you I mean you can basically check using the structure itself and then see like between change which residues making basically contact, right? Using biopython or like pymol
Hmm.
or maybe like chimera X basically. Like
Timer, yeah.
just checking like which residues are like making or like be with certain distance from the target residues. Like let's say 3.5 angstrom, how many are making those kind of like contact
I see.
basically. Yeah. So maybe I don't know like because MD is also like heavy, I think, right?
Yeah, yeah, it's pretty heavy.
Yeah, 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 yeah. So
But
so
I plan to
it
do it only on the top three.
oh, okay. I think it's good. I mean, to in my opinion, I see MD as a supporting way to explain things that I might not, like be able to explain. Right?
Hmm.
Let's say like with the same, like maybe like you want to explain the K like affinity, let's say later. You can use MD maybe like to see like how they sample over time, whether there is like crazy movement or something like that. So usually like I mean in my flavor, I usually use MD to explain something that I cannot explain from like experiment. Yeah,
All
that
right.
experiment cannot prove basically. Yeah, something like that.
All right.
Not yeah, yeah.
How about the numbers of designs that we have to express in the wet lab, Kak? Like
Well, again, it depends on your resource,
my
right? Like
my
how much money
plan.
you have.
So our plan was we got 90 kilo base pair of plasmid, from the iGEM. My
Like
plan,
one plasmid. I, oh,
um,
that in total 90 kb. Okay.
yeah, the DNA that they want us to, they can synthesize basically.
Oh, I see. I see. I see.
Um,
Um.
my plan was to be to express each. So we got three protein, right? In each protein, we got three designs on different positions. My plan was only select each design and each position just one. Is that
Oh, I see.
a flaw? Just one to
Well,
express.
yeah. That's to me. That's very confident, right? Um.
Mm,
Because I mean, again, if money is the, I mean, that's the limitation, right? Like, because you don't have like
mm,
infinite resource. I
mm,
think that's the right way to do. Like, you select the best,
mm.
right?
But you
Again,
meant if it is on it sits on the top one design, it does not mean the best in the wet lab from your experience. Like maybe
uh
you got the best results on the wet lab on the maybe third best on the computational simulation. Is
well,
this?
so far I've never ranked my design. I just select the, like the,
Yeah.
like top 200, let's say, that pass the filter. So I don't know like how you rank them. Maybe you make like a scoring function, but I've never done that, so
Mhm.
I don't know. I don't know like in my experience. Yeah, maybe I can do like later in my case, do like basically retroplastic analysis basically, to see like whether, like the metrics, computational metrics, really relate to the success in the wet lab, right? But
Yep.
again, sometimes it's really hard to tell. Yeah, yeah,
So
yeah,
I think you're suggesting pick more than one in each design in each.
yeah, yeah. It's just to lower the risk, right? I mean, if you want to get like, hits basically. Yeah,
Okay, got it.
yeah. If money is not the issue, or like resource is not the issue, right? Because I think time is more precious than risk. Like, like, because let's say
Yeah.
like if you can accommodate more design, then just do it rather than like doing one and then you realize, oh, it's not working. Order another gene, wait maybe another few weeks, right? And then you have like those kind of like waiting time that you kind of like, I don't know, waste maybe. Yeah. So it's better just to, yeah, test maybe more. But again, like, yeah, I think you have your mentor, maybe discuss with them as well, because in our lab we always test at least 96, well, like in 96 well format, right? 96 designs basically, at least.
At least.
Yeah, yeah, yeah. High throughput, basically, like we use high throughput, right? Like as I basically, like presented. Yeah, yeah, yeah,
All right. Well noted.
yeah, yeah. Okay. Is
Thank you so
there?
much. I think
Yeah, we can. I mean, we can always schedule, like another session, but I think maybe today it's a little bit late here as well. You can, if you have question, you can send me an email. I will try my best to answer.
okay.
Yeah.
Maybe before we wrap it up,
Mm-hmm.
mm, are there any question maybe from Kairman from the web lab? Yeah, or any other guy? By the way, Kairman is already graduating right now, so he is one of our seniors.
Oh, okay.
And right now, he is searching for maybe master's chance or
Uh-huh.
internship chance.
Okay, okay.
If
Well,
you,
good luck for that. Yeah, I
yeah.
mean, I remember like when I graduated. Yeah, I mean, I was happy, but
Mm,
yeah, I mean, I was like also like, in the search of like, you know, for graduate study basically. Yeah, yeah. Yeah, so I think that's all. Like I think, yeah.
All right, maybe one more, maybe for whoever in this meeting that that want to network with Kak Bima.
I think the story of Kabima was very inspiring, considering your background is from the physics, and I'm currently studying computer science, so I'm considering that we can pivot as a, you know, other way
Biologist.
to proceed in our career.
Yeah, I think again, it's all like depends on like what you are really interested in, right? So, well, I don't think so far like I have found like what I really want to do, the like specific field. I mean, the fact that I'm still like basically wandering to a new field that I want to basically find what I want to commit when I started my own basically, let's say like if I become like a PI or like a research scientist in a company. Yeah, I think that's basically like a fun thing to think about basically in your career. Like don't just be bounded, like be bound by one type of, like career choice. Obviously, like you want to get a job, right? But I think opportunities are like endless out there. So if you really want to do something that you haven't tried, I mean, just go for it. Try. I mean, there is nothing wrong in trying. You either fail or like like successful, right? So what's the cause of being, of failing? Nothing. It's just like, fail. Okay, life go on, and then search something else. Yeah, something like that. Yeah.
Yeah, that's really cool.
But I think, yeah, I mean, I'm amazed with you guys. I mean, doing well. I never imagined like when I was an undergrad doing like, protein design, right? I mean, I didn't I didn't know anything about, like research. I knew about like some research in physics and semiconductor, but not protein design. And then in fact, like protein design is, I think, one of the frontier right now, like especially after AI stuff. So it, I mean, like you guys are like basically like very advanced. I think it's just like, um, I don't know, like whether I don't think like people are doing protein design in Indonesia right now. I don't know any faculty are doing that or not. You can tell me if there are any faculties doing protein design. I would like to connect with them basically. But so far that I know, maybe not nothing, right? I don't know. Like if there are like professor are doing like the Novo protein design based on like what Baker Labs is doing basically. So yeah, I mean, you guys are like basically well ahead of, everyone there. So yeah, that's like basically like already like a big achievement, like understanding these pipelines. I didn't even touch or like know about this pipeline until like until the end of my PhD, right? When I was trying to find what kind of like science I want to do next after my PhD. So you guys are really like basically like ahead of, like many people. So I mean, keep doing that. If you want to, like continue your study, go for it. If you want to be in academia, but if academia is not your, like calling, do something else, right? I mean, yeah. But I think, yeah, iGEM is a good experience. I mean, I think if I knew iGEM when I was an undergrad, I would join it. But at the time, I didn't. I was not interested in biology basically. So yeah, yeah,
Right.
yeah,
There's one more from Kak Herman.
yeah, yeah.
Yeah. Sorry, Kak Bima.
Yeah.
My microphone still not working on the laptop, but I I must use two devices on it, and but before the question, I have a several question, but can I use Indonesia to speak
Yeah,
more clearly?
yeah,
Can
yeah, yeah.
okay. Sebenarnya saya punya dua pertanyaan Kak terkait
Mhm,
yang tadi desain de novo sama juga tentang DNA origami yang mungkin saya dahulukan tentang de novo protein itu sendiri di mana sebenarnya saya mungkin pengalaman tentang desain protein sendiri itu di kampus juga apalagi di Brawijaya itu memang lebih ke arah desain epitop vaksin seperti mungkin udah mungkin sudah banyak juga dilakuin di Indonesia seperti itu yang dimana kita memprediksi dengan menargetkan beberapa antigen tersebut gitu. Tapi kan untuk desain nanobody ini kan mendesain dari menyesuaikan dari protein yang kita ini kan habis itu dirancang residu asam aminonya sehingga membentuk suatu struktur protein baru. Nah, itu
mhm,
kayak kan mungkin itu juga hal yang baru ya dan juga mungkin kayak menanyakan terkait ada nggak kayak parameter yang mungkin bisa bisa di transisi yang dimana kayak krusial supaya kayak dari dry lab-nya sendiri itu bisa hasilnya itu bisa di eh, implementasikan ke wet lab. Jadi hasilnya itu maksudnya kayak nggak berbeda signifikan yang dimana mungkin dari dry lab mungkin kan secara algoritma ya kayak di ininya prediksinya gitu. Dan wet lab-nya kan mungkin dari di lapangannya kan juga pasti akan berbeda karena kan di wet lab-nya sendiri itu kan akan banyak human error dan lain-lain gitu. Faktor kondisi suhu itu kan juga berpengaruh di dalam mempengaruhi dari struktur si protein itu sendiri gitu. Gitu.
mhm,
Nah, itu kayak yang kedua sih terkait sebenarnya saya penasaran juga yang tentang DNA origami di mana kan kok DNA origami itu prinsipnya juga melipat 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 DNA sesuai dengan bentuk dan struktur yang kita inginkan. Di mana juga sebenarnya saya kayak memikirkan satu topik juga terkait ini yang dimana definisinya itu mirip sama namanya itu aptamer gitu. Aptamer itu
mhm,
juga definisinya juga sama melipat menjadi 2 dimensi dan 3 dimensi untuk bisa sebagai sensor dan
mhm,
bisa sebagai terapeutik gitu. Akan
mhm,
tapi kalau dari aptamer itu sendiri sejauh yang saya baca dari beberapa paper itu memang mengatakan bisa sebagai terapeutik tapi tidak sepenuhnya bisa dan masih lebih ke banyak sensor karena dari si DNA itu sendiri ketika dilipat itu mungkin kurang kurang mengikat si protein yang ditarget itu mungkin kurang ya sehingga menjadi kayak afinitasnya atau ikatan interaksinya itu jadi rendah di mana tidak bisa sebagai terapeutik karena kan kalau misalnya sebagai terapeutik kita harus bisa memblok active site dari suatu protein target kita. Nah,
mhm.
kalau misalnya DNA origami ini apakah akan lebih baik apakah ininya lebih baik dari aptamer itu sendiri atau sebenarnya definisinya sama, cuman kayak maksudnya kayak aptamer itu bagian dari DNA origami itu sendiri gitu.
Iya ya, ya makasih Herman pertanyaannya. Jadi kayak yang pertama, kalau menurut saya tuh sebenarnya apa yang kalian lakuin itu udah benar gitu dengan apa namanya me, melakukan filter beberapa uh, matrix dari prediction itu kan gitu. Cuman lagi-lagi kayak nggak terlalu susah aja untuk memprediksi bahwa apakah memang korelasinya 1:1 gitu kan antara antara filter di silico, in silico atau dengan apa yang nanti hasil dari wet lab itu gitu. Karena di sini aja kayak nyoba misalnya 96 desain gitu, kemungkinan kayak dapat hits misalnya lebih dari 50% aja itu udah kayak bagus banget gitu, kayak 40 hits gitu. Nah, apalagi kalau misalnya kalian nyoba hanya satu kan gitu kan, kayak kalau misalnya hitsnya 1%, oke, bakal berfungsi itu proteinnya gitu kan. Cuman kan kayaknya kayak terlalu riskan aja gitu untuk cuma rely on that design and hope that it will work gitu kan, kayak gitu. Jadi kayak aku bicara kayak tes mungkin beberapa desain untuk untuk nurunin resikonya. Jadi kalian kayak beneran kayak oke, dalam rentang waktu yang sama ngetesnya gitu kayak bisa langsung dapat 5 hasil, itu kan lebih baik dibanding kayak dalam waktu yang sama cuma dapat 1 gitu kan. Dan korelasi antara matrix in silico sama wet lab itu mungkin ada, cuman kayak susah aja gitu, misalnya top 10 gitu, kayak itu kayak susah aja prediksi yang mana nanti wet lab yang bakal jadi yang terbaik gitu. Bisa aja kayak yang nomor 3 lebih baik dibanding yang nomor 1 kalau di wet lab dibanding dengan in silico gitu kan, kayak gitu. Jadi itu menurutku ya gitu. Maksudnya bisa aja salah, tapi kayak itu menurut pandanganku gitu. Tapi yang kedua, aptamer, ini sebenarnya DNA origami itu kan bagian dari DNA nanoteknologi ya. Aptamer itu juga bagian dari DNA nanoteknologi, dan kenapa aptamer lebih banyak sensor, karena memang untuk bikin aptamer pun sekarang kayak orang masih nyoba dengan apa namanya, memproduksi DNA atau RNA library terus dites ke target proteinnya gitu kan. Dan seperti yang Ewan bilang itu benar, kayak ketika dia apa namanya, bind itu, apa sih bahasa Indonesianya, nempel ya, atau interaksi dengan protein target itu, mungkin dia tidak sepenuhnya apa namanya, menghalangi active site atau epitope itu gitu. Jadi kayak protein lain yang target benar itu masih tetap bisa gitu untuk ngedisplace si aptamer itu gitu. Jadi kayak sebenarnya untuk therapeutic itu kan semua perihal relatif ya gitu. Jadi kayak yang mana yang bisa bind lebih kuat dibanding yang lainnya gitu kan. Jadi kayak untuk ngeblok apa namanya, signaling selanjutnya gitu. Jadi kayak dimana kalau kita desain binder atau therapeutic binder itu kayak kita memang benar kayak pengen affinity-nya tuh kayak sub nanomolar, di picomolar range gitulah. Jadi kayak biar dia memang beneran kayak potent dan efisien gitu dalam ngeblok active site-nya itu gitu. Sedangkan aptamer nggak tahu, kayak aku nggak begitu ini berapa affinity-nya dan apa namanya, binding antara DNA RNA dengan protein juga nggak yang kayak protein dengan protein kan. Jadi kayak make sense kalau dia nggak yang terlalu ke therapeutic gitu. Tapi untuk sensor memang aptamer juga apa namanya, banyak yang pakai, terutama untuk detect apa namanya, reseptor protein dan apa namanya, untuk analisis apakah ada protein tertentu di cell surface gitu, misalnya kayak gitu.
Soalnya juga dari aptamer itu sendiri kan juga sejauh pemahaman saya itu juga dia DNA-nya nggak begitu panjang, dan juga kebanyakan saya membuat kesimpulan dasar singkat gitu ketika membaca beberapa paper itu, juga dia pelipatannya itu lebih mengarah ke arah struktur 2 dimensi, struktur di dua dimensi, kayak soalnya seperti 2 dimensi, apa seperti bentuk hairpin atau dan lain-lain gitu. Tapi kan kalau misalnya DNA origami ini melihat dia juga bisa dibentuk struktur 3 dimensi, itu kira-kira udah banyak nggak yang kayak pengalaman dari DNA origami itu, kayak di mana penelitiannya dia membentuk suatu struktur 3 dimensi di mana dia menyesuaikan dengan si active site si protein itu, di mana melihat affinity-nya itu lebih kuat nggak gitu dengan bentuk struktur yang variasi struktur yang di ini.
Ya jujur kayaknya kalau buat DNA origami itu lebih ke platform untuk dia itu ningkatin. Jadi kayak bukan langsung kayak bikin DNA origami yang bisa kayak apa namanya complementary dengan target epitopnya gitu atau target dari si protein target itu gitu. Karena kayaknya untuk bikin, maksudnya protein target itu kan kayak surface itu kan kayak topologinya kan apa namanya kompleks ya gitu. Jadi kayak kalau menurutku kayak DNA origami kayaknya nggak bisa untuk se-accurate itu gitu. Jadi kayak orang pakai DNA origami biasanya untuk naikin, let's say like let's say kita punya DNA origami yang ukurannya yang bentuknya rectangle gitu kan. Jadi kita bisa pakai itu untuk sebagai platform vehicle untuk lebih banyak apa namanya naruh molekul binder yang akan bind ke target protein itu gitu. Jadi kayak kita increase local concentration-nya, jadi kayak lebih efektif aja untuk bind ke target proteinnya itu gitu. Jadi kayak DNA origami kayak istilahnya kayak 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 framework dari si antibody itu. Nah nanti CDR-nya bisa aptamer gitu. Kayak gitu analoginya. Gitu misalnya gitu. Jadi kayak dia lebih tinggi affinity-nya karena dalam satu DNA origami kita bisa punya 10 aptamer yang sama yang mana local concentration bakal lebih tinggi. Kalau local concentration lebih tinggi, dia bakal afinitasnya lebih tinggi juga kan. Karena dia bakal kooperatif aja kan. Kalau satu misalnya aptamer bind, terus yang satu ini dia tiba-tiba dissociate. Nanti yang lainnya mungkin bisa langsung cepet nge-grab itu lagi gitu. Untuk bisa tetap stay di sana gitu. Untuk ngeblok gitu misalnya. Kayak gitu. Dan mungkin dari segi geometri juga benar itu bisa bikin dia ngehalangi target protein itu, tapi kayak untuk bisa bikin shape complementarity yang beneran complementary itu kayaknya DNA origami nggak yang sampai sana gitu. Jadi kayak dia mungkin bisa ngeblok gitu, kayak istilahnya kayak blok beneran kayak pagar gitu, tapi nggak yang bisa kayak blanket yang gitu. Karena untuk bikin curvature yang dengan radius yang 1 nanometer itu kayaknya nggak terlalu, maksudnya mungkin sangat challenging sih gitu untuk DNA origami gitu.
Berarti DNA origami ini juga kalau misalnya saya bilang kayak mungkin apa ya bahasanya, mungkin kayak kalau di nano itu kan kayak ada matriks, ada yang kayak biasanya kalau ini kayak saya bahasanya enkapsulasi ya. Jadi kayak ada matriksnya, ada wadahnya, ada isinya. Jadi kayak DNA origami itu bisa kayak ibaratnya seperti wadahnya gitu ya, kayak
Iya, dia bisa jadi vehicle-nya gitu, platform untuk pack more molecules on it gitu, or inside it gitu. Kayak gitu.
Oke.
Dan itu juga bikin molekul itu mungkin lebih resisten terhadap apa namanya all sort of like enzyme like protease or endonuclease misalnya kayak gitu. Karena kan dia kan bulky kan. Jadi dia bakal give protection lah, like longer than if you only deliver specific aptamers that may easily be degraded. Like if it's just a single, like small DNA or RNA gitu, kayak gitu. Jadi ya, maksudnya komplementari sih apa namanya fungsinya, cuman ya gitu. Itu a nice platform dan apa namanya ya untuk apa namanya to have aja misalnya kalau ini nggak bisa, itu mungkin bisa dilakuin gitu kayak gitu. Ya.
Oke deh Kak, terima kasih
Ya.
banyak atas informasi.
Iya.
Oke, Kak, thank you so much.
Iya.
Gak enak udah lama banget. It's longer than we expected. I think we have to cut it off here.
Iya. Makasih banyak juga udah ngundang. Mohon maaf kalau misalnya belum bisa menjawab semuanya. Kita sama-sama belajar ya. Jadi kayak kalau misalnya ada pertanyaan, shoot me an email. I'll try my best to answer.
Oh yeah. By the way,
ya.
um, considering our conversation, kalau kakak jadi one of our advisors, bersedia nggak ya kak? So, jadi kayak di Ajem tuh, there's two advisors, satu itu PI. PI itu yang lebih, ya just like PI, lebih formal dan lebih kayak dosen team lead gitu. Tapi ada juga advisors, nggak mesti dosen, tapi it could be maybe kayak kita coba sediverse mungkin advisorsnya dari berbagai aspek gitu. And it doesn't really require you to do anything sih. Kayak hari ini udah knowledge transfer aja, kayak you can count as one of our advisors gitu.
Okay. Maybe I'll think about it.
Yep.
Soalnya kayak aku nggak tahu kayak sekarang kalian udah berapa punya advisor dan siapa aja gitu kayak. Aku cuma kayak emang lagi-lagi kayak again, I cannot promise that I can always like be like how to say, like be there like in like basically promptly, right? But yeah, I'm always happy to share.
Yeah. It doesn't burden you like you have to meet with us
Yeah.
every day.
But I mean, because
I mean, it would be
it's
cool
another
to have.
responsibility, right? So
Yeah.
if I cannot make my, I mean, like like make it there basically, like if I have my responsibility.
Yeah.
But anyway, like I'm always happy to help. But
Yep.
Thank
But
you, Vincent. Everyone.
Yeah. If you do so, you don't really.
Selvani, right? Selvani.
Selvani,
like,
yes.
Yang email aku. Yeah.
Okay.
Yeah. Thank you so much, Kak.
Thank you so much.
Yeah.
Can we do documentation before, please?
Apa?
Dokumentasi.
Maksudnya apa?
Foto.
Oke.
Mungkin bisa aku bantu pandu ya teman-teman yang belum on cam, bisa nyala yuk. Kak Frederick, Kak Afif, Tiffany, dan lain-lain. Oke deh. Kalau gitu aku coba hitung ya. 1, 2, 3. Maaf lama aku sambil screenshot. Sekali lagi coba gaya simbio teman-teman. Kak Bima juga bisa dicoba jadi DNA seperti ini.
Oke.